Hello. Hello. Surprise! Oh my God, I'm such a dork. Hope you appreciate my dorkiness, guys. All right, so we are going to finish chapter 13, meiosis today. All right, proceed to the iPad. Share the screen. Go. Go. Oh my. Oh, come on, team. Team, help Dr. Keller figure our stuff out. All right. Sweet. All right. So we are continuing with chapter 13. So I just want to remind, I'm going to just use this blank page as a reminder. So if we have mitosis, and we have 1, 2, 3, 4 chromosomes, but 2 pairs, what we are going to see for mitosis is that those are going to each replicate to form our sister chromatids. We are going to get a mitotic spindle forming, and then we are going to get 2 identical cells out, both now. Well, here, I want you to see the separation. So in anaphase, these are going to start separating, and we get one little, one little, one little, one little, one big, one little, one big, one little. And then we are going to get our 2 cells, each now with a pair of long homologous chromosomes and a pair of short homologous chromosomes, right? That's mitosis. We did DNA replication, followed by separation of sister chromatids, all right? And so what we see with meiosis is that we're just gonna add a whole phase. So we start the same, all right? So we still starting here. For example, I always just do 2 pairs because it's easy to draw. And we're gonna do the exact same thing here where we are going to go through DNA replication. What phase of the cell cycle? That's S phase of interphase. And so we are going to end up with, I didn't draw that very well. I'm gonna make one of those smaller. We're going to end up with, oh my gosh, I'm getting all these alerts and what is that box? You can't even see the box. We're gonna end up with one, oh my gosh, something is weird. Okay, is 2 duplicated pinks and 2 duplicated blues, 1 big, 1 small of each, all right? Remember that at metaphase, they are going to line up so that what is going to split? We are going to see that at metaphase, or in meiosis 1, we are going to see the separation of homochromos, or homologous chromosomes. So again, if you draw that midline, you better be separating the 2 big ones from each other, the 2 small ones from each other, the 2 medium-sized ones from each other, etc. All right? And again, just to give you an example, that after DNA replication and at the metaphase, you can line up in different ways. All right? You're still going to get separation of homologous chromosomes. It's just how they line up, right? And those are both perfectly fine. All right, so when they split, and I'm not going to show anaphase 2 here, but what you're going to end up, and I'm just showing you both options, is that you would get all the pink in one cell and all the blue in the other, or you would get a mix, depending on which meiosis you were doing, which way they lined up in metaphase 1, right? They can line up in 2 different ways. All right, then what we're going to see is a separation of sister chromatids in this next division. So again, I'm going to draw my metaphase plate so that sister chromatids are going to split. So in this top cell, we would get, if they were lined up that way, we would get 4 cells, one with all mama, and one with all daddy, right? If metaphase 1 lined up that separate way, we would get 3, 4. We would get big mom, little, oops, sorry, little, big. All right. So we are going to get 4 haploid cells from meiosis, each with only 1 copy of each chromosome, right? 1 big and 1 small. In comparison to the original cells, we had 2 big and 2 small. All right, so that's just a reminder of meiosis. And sorry, I'm getting all these weird alerts. Okay, and so the last part of this is talking about ways that meiosis increases genetic diversity, right? So it does it in 3 main ways. The first is crossing over, and this occurs in prophase 1. Right? And crossing over is a way of exchanging genetic material so that you can increase your diversity. Independent assortment is what I've been talking about, how the chromosomes can line up in different ways at metaphase 1. Right? It's the random orientation of homologous pairs at that metaphase plate. And then, of course, random fertilization. So the fusion of 1 random egg and 1 random sperm during fertilization, right? So we're going to talk about each of those. First one is called crossing over, and it occurs in prophase 1. And again, in prophase, that's just right when we start to see those chromosomes condensing, right? So crossing over is a genetic rearrangement between non-sister chromatids, right? In prophase 1 of meiosis 1, right? It's non-sister chromatids, but they are homologous chromosomes, right? So let me see, I'm gonna just change my color here. So remember, so what we're starting with here is 1 pair of homologous chromosomes. They went through S phase to duplicate into these sister chromatids, right? So those are now duplicated homologous chromosomes, right? So here, these are the homologous chromosomes. Here we have the duplicated homologous chromosomes. Crossing over occurs between these homologous chromosomes so that dad's chromosome and the chromosome from dad and the chromosome from mom literally swap pieces of chromosomes, right? To give you what are called kind of recombinant chromosomes. I don't care if you know that word. What I do care is that the 2 chromatids that kind of get close to each other and they swap material, right? And it is called a chiasma. And that literally just means the X shape that occurs when crossing over events happen, all right? So we're gonna do a lab. Hopefully we're gonna have time to do some playing with pop beads so you can actually physically see how material can get exchanged, right? So that's one way to increase genetic diversity. Metaphase 1 is another place that we increase genetic diversity, and that's through this process of independent assortment, or in other words, how do you line up your chromosomes at metaphase 1? How do you line up your, I'm going to put homologous chromosomes, your homochromos at metaphase 1, right? Remember, each pair of homologous chromosomes can orient with either its maternal or paternal homologue closer to a given pole, right? So for example, we've gone over this in our drawings. You can have one possibility where all dad's chromosomes are on the left and all mom's are on the right, or you could have a possibility where they're swapped, right? So either way, what's absolutely essential here is that when you draw your kind of metaphase plate, that what you're going to see are homologous chromosomes separating from one another, all right? We're not getting sisters separating. You're going to see homologous chromosomes separating, right? And you can either line up in either of these orientations. And depending on which one happens, you create different haploid gametes, all right? So what you can see is after metaphase 2, you can get different combinations of chromosomes here, right? So depending on possibility 1 or possibility 2, you get different gametes. And imagine this is 2 possibilities, right? We have 23 pairs of chromosomes. So there's a huge number of possible ways that you can line up, each of those giving rise to different combinations of haploid gametes, meaning that the egg and the sperm, all the egg and the sperm in a person's body are completely different from one another, right? And that's why you might have sisters or brothers that look very much different than you, right? Because you are sharing the same DNA, but depending on, you might have gotten a lot more DNA from mom or from dad, depending on your, the way they lined up. All right, so the last one is random fertilization. So I think that this is an amazing kind of statistic, or I don't know, fact. So a zygote. So remember, a zygote is a fertilized egg. So egg and sperm have come together to create a zygote. A zygote is formed by the random union of gametes, right? Meaning egg and sperm. In humans, every gamete, right, egg and sperm, represent 1 in 8.4 million possible chromosomal combinations just from independent assortment, right? How they line up on that metaphase plate. There's even more genetic diversity on what genetic material was exchanged and crossing over, right? And then random fertilization produces a zygote with about 70 trillion different diploid combinations. So you truly are unique and one of a kind, 1 in 70 trillion. Just to give you a sense of that, all right? During fertilization, I just want to remind you, you have a female gamete, and this N means how many copies of each chromosome, right? So haploids have 1 N, whereas, so those come together. When the male and the female gametes come together, you get now a diploid zygote, and diploid means you have 2N, or 2 copies of every chromosome, right? And just to give you a sense, this here is an egg that's trying to be fertilized by many, many sperm. And eggs have all these amazing ways to prevent multiple sperm from getting in, right? So during fertilization, and I'll show you this movie during class, hopefully, remind me, they have this really cool way that sperm get into an egg. So here is our little sperm over here, and it's coming to the egg. And the egg has many different layers of material, and the sperm actually releases enzymes in kind of its little head region that will eat through this kind of yellow kind of coating on an egg. All right, and then eventually when the sperm, after the sperm does that, it hits kind of the actual egg, and the sperm just releases its nucleus, all right? Just a nucleus. So it's not actually, the whole sperm is not actually entering, it's just having the nucleus, all right? And then upon fertilization, the egg undergoes a lot of dramatic changes to prevent polyspermy, or to prevent multiple sperm from coming in at one time, which could be very detrimental. All right. I think this is the last thing. So yeah, the last thing, I just want to point out that gametogenesis, or the making of gametes, and that is all again through meiosis, can occur in different ways, whether you are making sperm, for example, this would be in males, or if you're making eggs. So that would be in females down here, all right? So I'm gonna look at just the top half right now. So it's called spermatogenesis, All right, so spermatogenesis, the making of sperm, going through meiosis to make sperm. So we start again with a diploid cell, all right? It goes through DNA replication. So don't forget, we have to go through DNA replication always. Then we have prophase 1, where we start to see the individual chromosomes. Here we have metaphase 1, where the homologous chromosomes are lining up. Here we have anaphase 1, and here is where we're getting homologous chromosomes separating. All right, then we have our telophase 1. And then in meiosis 2, we are getting, this is showing anaphase 2 and eventually telophase 2. And here we create 4 different unique haploid sperm, all right? And that uniqueness, again, comes from crossing over in prophase 1 and how those chromosomes are lining up in metaphase 1, right? In oogenesis, some people say oogenesis, but I just can't. I mean, it's too weird. So I say oogenesis. Actually, instead of making 4 cells, eggs need a lot of good stuff in them to support the growth of a new organism, right? So what eggs do during oogenesis is that they do the same thing. You have, DNA replication. They have prophase 1, where we're going to get crossing over. We have metaphase 1, where they're going to line up on that plate. We again have anaphase 1, where we get homologous chromosomes separating. And then we get, anaphase 2, where we get sister chromatids separating. But instead of getting 4 haploid cells, this in an egg, you are going to get 1 big, meaty, strong haploid cells. And these other ones are just going to go away. So they kind of, all the good stuff gets absorbed from those little cells that are just going to degenerate. And they become what are called polar bodies, almost like little scars for where the egg was. All right. So that's all about meiosis. Remember, there are lots of practice questions that you can do. You will have to draw things out on this test, so be prepared for that. Other than that, come with questions, please, to class. Peace. Bye. Ciao. Adios. Toodles, poodles. Ta-ta for now.